The next talk will be by Bruce Menge. Bruce is a distinguished professor of zoology at Oregon State University. His major research areas are marine community and meta-ecosystem ecology, physiological ecology, and he is currently focused on the impact of climate change, including ocean acidification, on coastal marine ecosystem. Bruce is the lead PI of several very large programs. One is the PISCO, which we'll hear about today. The PISCO is a partnership for interdisciplinary studies of coastal ocean. Another one is OMEGAS, which is the Ocean Margin Ecosystem Group for Acidification Studies. And another program, ACIDIC, which means Algal Community in Distress, Impact and Consequences. Bruce is a fellow of the American Association of Advancement of Sciences and member of several distinguished societies. Among his honors, he got the George Mercer Award, honorary doctorate of science from the Long Island University, and the Western Society of Naturalists Lifetime Achievement. Bruce, please.
Well, thank you. It is, as everybody has said, it's a real pleasure being here. This is my first trip to Israel, and I've been having a really good time interacting with people and seeing a little bit of Israel, at least the inside of conference rooms at the various places that we've gone to. So PISCO, you saw the first slide. The focus of PISCO is what we called, so Nancy, did you kill my pointer? Oh boy. We called this a big, hairy, audacious goal, and the idea was to understand the dynamics of a large marine ecosystem, which is this region from along the U.S. West Coast. This is called the California Current Large Marine Ecosystem. It's one of 64 around the world. And we were interested in answering a couple of some questions, two big goals. Oh, another one. Okay, got to back up. The first goal was to figure out how this system works across multiple scales, and is it changing? And then the second major goal was to use this knowledge to improve public policy, inform and improve public policy. So PISCO is a consortium of scientists from 4 different institutions. The goals, again, were to understand the dynamics of this system, to train a new generation of scientists, especially trained in interdisciplinary approaches. And one of those people who has come through our lab is here today. It's Gil Reloff, who was a postdoc with us for 3.5 years and is now here in Israel trying to use a similar approach to understanding the coastline of the Mediterranean coastline of Israel. And then finally, to share this knowledge, hopefully to influence science-based decisions and get them made, hopefully, in the right way. So the elements of PISCO include these. We do monitoring at an ecosystem scale. We train and encourage the use of interdisciplinary ecosystem science. This involves generating enormous amounts of data, and so we also have a program of data archiving and sharing. There is a major amount of outreach to public and decision-making user groups. There is this interdisciplinary training, which actually involved offering courses to graduate students and postdocs, and actually even some professors took the courses. And overall, this included required coordination of a distributed research team. We have teams of people at these different institutions all ideally doing things in the same exact way. So there are 12 PIs. Among the different types of people, in addition to the lead scientists, are a PISCO-wide coordinator, somebody who is basically helping us to all stay on the same track and in communication. Science coordinators who oversee the field teams and the activities in the field. Policy coordinators who are responsible for communicating with policymakers and managers. We had database support staff to keep the database operating. And then the sort of the heart of the activity is postdoc students, research interns. These are undergraduates who come in and work for a summer to help with field activities. Technicians who sort of are the leaders of the different field crews. We also had an executive steering committee, which this was a subgroup of the PIs, which sort of from each campus that oversees activities on a regular basis. And we also had have an external advisory board. These are scientists from around the country who come to advise us and give us feedback about how we're screwing up and whether or not we're doing things that make sense. So the expertise that we have ranges from ecology and oceanography to biomechanics, genetics, molecular and organismal physiology, biogeography, and then through collaborations we've also involved various socioeconomic folks and fisheries people. And this is a photo of the PIs taken a few years ago. My wife, Jane Lubchenko, is right there. She and Steve Gaines, who is right there, and I were the three PIs that got the ball rolling in this effort. And as one indication of productivity is that we've published, it's actually now more than 417 papers since the program started in 1999. So the overall focus of our activities is to learn about how coastal ecosystems can be sustainable. And we see that there are three major factors or three major research themes that feed into this very large issue. Climate change sort of overarches everything because that's going to influence basically everything. We've had a lot of activity with regard to marine reserves and with regard to marine resources. So I want to run you through a few examples of some of the research activities, and then I'll talk about some of the other aspects as well. So the core of PISCO is the long-term large-scale monitoring. The idea that we sold to the funders, which were at least 50% private foundations, was that to understand systems like this, you needed to sample both at the scale of the system and to do it for a long time. The only way you're going to be able to assess any kind of effects of climate was to do it for as long as possible. And as you all know, doing long-term research is not something that federal agencies like to participate in. So we had to be creative, and that's why we approached foundations with this idea initially. So this just shows some examples of our biodiversity sampling program. We basically go out and run surveys. Let's see, what's next? We run surveys that essentially quantify biodiversity. In other words, we count and estimate the abundance of every single species, at least macro species that we can see in the field. Things that are harder to identify, we try to bring back and identify when we get back in the lab. But even some of those are hard to identify without using genetic approaches like Nancy just summarized. So we're not actually counting all of the species, but we're getting hopefully a large fraction of them. And then the long-term aspect is shown here. This just basically shows some of the different monitoring activities we've done. We measure recruitment of key invertebrates and fishes. We look at nutrients and productivity in the inner shelf region of the coast, and we also sample using moorings. And so these are color-coded. The red shows the recruitment time series at the different locations along the coast. The oldest one actually goes to 1989, which is started up at Oregon State by me. And then we started sampling the water up in Oregon and eventually down further south for phytoplankton, nutrients, and other things. And then the mooring programs were started with the beginning of PISCO, and there are 3 of them, one in Oregon, one in Central California, and one in Southern California. As an example of what kinds of patterns you can get, this shows the results of the water sampling program, which is taken basically by just going out to the rocks and sampling the water right next to the rocks. So we do take oceanographic, in other words, sampling further out from moorings or from small research vessels, but this is taken right along the shore. And it shows the data since it started in 1993 over the region from northern Oregon down through northern California. And the basic point is that it varies in space and time. There's temporal changes you can see as you follow the different colors through. There was a period of high productivity here in the early 2000s up in this region. Another one here. Some sites are very unproductive, and they are that way almost all the time, with occasional blips here and there. So this is important stuff to know, because as I'll show in a second, this high productivity area has some important implications, like this. This is mussel recruitment taken since 97 up to 2005. And again, over this is the whole range from Santa Barbara up through northern Oregon. And again, this is color-coded, so high recruitment areas you see for mussels are up north. You can see the seasonality in this very clearly. You can also see that there is temporal variability, and there's also the spatial variability from north to south, but also within regions. There are some sites that have higher recruitment, others that have lower. Notice that the high recruitment site also is where we saw the high phytoplankton abundance site. Now, these are patterns of recruitment. We go out and sample every month and count what we get. Over here is shown what we think is the interpretation of these spatial differences, at least. This shows an analysis done recently, led by a former postdoc with the PISCO Consortium. And what it shows is the geographic location of oceanographic fronts up and down the coast. These are areas where different masses of water meet. They tend to accumulate larvae and often attract fishes and other things that are interested in eating the larvae or eating the zooplankton. And what he found in this analysis, which used oceanographic and the ecological data like this, looking at fishes and invertebrate recruitment, was that these fronts tend to be spatially variable along the coast. Along the Oregon coast, you have a high proportion of persistent, where is this scale? Well, what the red means is lots of fronts and they are persistent, and the cooler colors mean there are relatively few fronts, and if they form, they don't last. So you can see that the high frequency of fronts that occurs up in Oregon is associated with these high levels of chlorophyll and high recruitment rates of mussels and also of barnacles. We also do monitoring in the subtidal. This is where the sampling has been done. There has been some sampling in Oregon. It's tougher to do because it's a more challenging environment to work in. But this is a very extensive sampling program. These, again, are all sites that are monitored. And here we also do abundances and distributions. So kelp density, invertebrate abundances, density and distribution, and fishes. So I want to run through fairly quickly some successes that we've had. And again, these are things that that have already or are hopefully will be useful in carrying scientific results to policymakers. And the goal with PISCO, one of the goals was to bring scientific knowledge as quickly as we could to policymakers and managers so that they could use this information in their decision making on things like marine reserves and so forth. So we've had some discovery successes. We figured out, discovered and figured out why dead zones began to occur along the coasts of Oregon and Washington in the early 2000s. We have, through our long-term monitoring data, we've been able to relate certain ecosystem processes to climatic patterns. We've done various kinds of assessment things, so we are currently looking at the biological responses of different key components in these systems to ocean acidification. And we've also had a lot of influence on influence in discovering how marine protected areas function in networks. We have formed partnerships with a lot of different people, just a couple here. One is with the Ocean Observing Network. We do our observing close to shore. Our deepest moorings are about 30 meters deep, and we sample out to about 100 meters deep, and that overlaps with what the OOSs do, which is to sample further out on the shelf from roughly 50 meters to hundreds of meters depth. We also have done monitoring in relation to reserves and sanctuaries, so we can compare the patterns inside and outside of these places. And with regard to policy, we've been heavily involved in the design of marine reserves and in their application, and are currently heavily involved in marine spatial planning with a special focus on catch shares as a way of managing fisheries. So hypoxia. This is something that first was reported in 2002. People went out to the beach, people including us and our crews, but also citizens, and they saw pictures like this, beaches littered with dead fish and crabs and other sorts of things, or areas on rocks where a whole mass of crab carcasses just sort of were accumulated by waves and the shape of the rocks. And because PISCO existed, we were actually able to respond to this almost immediately. We saw that there was something going on. We didn't know what it was. We could generate hypotheses and ideas and then act on those ideas basically right away, which is what we did. So we started sampling oxygen because we felt that might be important, and it turned out that was the issue. This map shows the region of the Oregon coast that was hypoxic in, when was this? I think it was 2005, but I'm not sure. You can see that the red area is very low, or actually high oxygen. The blue is the area of low oxygen. And this mass of water, which comes in at depth, will eventually reach shore if there is strong upwelling that brings this low oxygen water to close to shore. And the other thing that this work has done is to catalyze a lot of other research efforts, especially amongst oceanographers at Oregon State University and at the University of Washington. So here's just some data showing the progression of hypoxia. The blue dots, each one of these dots is a single oceanographic water sample taken from mostly ships. And oceanographers have been very busy along this coast, starting in 1950 when the Oregon State University Oceanography School was started. These kinds of samples were taken, and you can see here, so this is depth from surface water down to depth, and this is the amount of oxygen in the water. The blue dots are all the data from 1950 to 1999. And this was pulled together by one of my research associates. He was a postdoc with us. He's now a research associate. His name is Francis Chan. And you can see that as you get to the surface, the oxygen levels get to be high. They are very low at great depths. This is a very typical oceanographic signal. This red line shows what happens. This is hypoxia. This is the level of oxygen that distresses and kills organisms. Right at 0, of course, is anoxia, which means no oxygen in the water. So in the early part of the 2000s, we saw these green samples, and these are now mostly our samples, the green ones. And you can see that for the first time, we're seeing very low oxygen at shallow depths. In 2006, which is still the worst year that we had, we saw no oxygen in many, many samples, again, in shallow depths. And the pictures I showed you earlier of dead organisms like these are magnified. We have taken video transects of the ocean bottom, and it's just basically a sea bottom of death. Everything is dead. Fish are dead, various invertebrates rotting in place, and so forth. So this was covered quite a lot. This was one of our major outreach efforts. These are flyers that we created and made available to the media and to policymakers and so forth to explain what this meant and how it might have happened. We got attention in sort of a step function way, especially after the 2006 event. We got a lot of media coverage. So moving on to the climate impacts, again, we have these data sets. This one looks at mussel recruitment, which we've had going at least a couple sites since 1989. This covered a period of 20-some years, and so I began looking at how this related to various climate cycles, supposedly natural ones. The ENSO cycle, El Nino Southern Oscillation, is one of these. The North Pacific Oscillation is shown here. This is a newly discovered one. It's mostly related to variations in currents along the shore. And the Pacific Decadal Oscillation is a third. Now these have various different time frames. El Nino is on a 3 to 7 year cycle. The NPGO is on a 10 to 15 year cycle. And the Pacific Decadal Oscillation is 20 years plus. So what I did was I just related the abundances of mussel recruits to two things: this North Pacific Gyre Oscillation, which you can get measures of on a monthly basis, and the abundance of phytoplankton from these surveys that I showed you earlier, and just ran it through a multiple regression, and what I got was this kind of pattern. That mussel recruitment is responding to two things. This is basically a proxy for food abundance. Mussels are filter feeders. And this is a proxy for currents and mostly currents. And you can see that from the tilt of the plane here that the recruitment that we were seeing was being driven in part by these two factors. And in this case, the R squared was around 0.8, I think. So a very strongly explanatory relationship. Moving up a bit, we also have looked at mussel growth through time. We put out mussels and measure their growth on an annual basis. So I did a similar analysis with that and found that in this case, it related to the El Nino Southern Oscillation and the Pacific Decadal Oscillation cycles. Again, in a complex three-dimensional sort of way, both food abundance and in this case temperature, which is is influenced by both of the ENSO and the PDO sorts of effects. So chlorophyll and these effects through their influence on temperature are driving variation in mussel growth. And this just shows how it varies with the different cycles. growth is faster with El Nino and slower with La Nina, and faster with warm PDO and slower with cool PDO. And what the analysis shows is that the primary driver of growth is temperature. Mussels are responding mostly to temperature and affected about at a 20% level by the amount of food availability. The other CO2 problem is, as Nancy mentioned, is ocean acidification. And it turns out that as a result, this was sort of suspected for a long time by oceanographers that we would experience relatively acidic waters being brought up to shore because of upwelling. It was actually documented by a paper in 2007 by Richard Feely and his colleagues at NOAA. And what they found was that during a cruise, which began up north and went south, so this is basically a snapshot, a slow-moving snapshot down the coast. The red shows the depth from which water that is reaching the shore comes from. And the interesting thing is that highly acidic water is shoaling along much of the Oregon coast, along the northern and central California coast, and down here in the San Francisco Channel region, and then further south. So because we were involved in PISCO, we were able to mount a project and get additional funding to study this problem. So one of the big issues with ocean acidification is that there is a massive lack of data. We don't really know, except for a couple of places, one in Hawaii and one in Monterey Bay, what the patterns, what the time series patterns of ocean acidification are. What are the pH levels in these locations? One problem has been that there weren't available instruments that you could deploy in the ocean and expect to have work. The technology wasn't really developed until about 5 or 6 years ago. And the initial machines were very unreliable. They involved moving parts and squirting reagents in and out and then sensing the data or logging the data. In about a few years ago, another device was invented. This measures pH only, but it's a solid state machine, and it's based on basically information you can get off the shelf, or at least you can send to Honeywell and they will send it to you. This is called, the instrument, the heart of the instrument is called a Durafet, and we had marine engineers work with us to build these instruments, which here's the sensing end here to log pH. We've also got these kinds of instruments out on our moorings just offshore, and so we're measuring pH intertidally and in the shallow subtidal. We also, on the moorings, but not in the intertidal, are measuring CO2 using these more complicated machines. And this map just shows where the sensors were located in the project that we call Omegas. The red dots are where sensors were both intertidally and subtidally. Another grant has added these 3 yellow dots, and if we get funding for a continuation of the Omegas project, we'll add the black dots. So what we're working toward is developing a network of sensors that will give us data, and I'll show you an example of what we got from before. Now, PISCO is a research consortium, and again, it was created because we felt the way to study these kinds of systems was to do it in an integrated way at the scale of the system and through time. So this slide is making a pitch for the consortium approach to studying these kinds of very large problems. PISCO is the core group upon which the others are based. Omega's added people from University of California, Davis, that are at Bodega Marine Lab, Monterey Bay Aquarium Research Institute. And the work that we're doing basically uses PISCO approaches. So we do intertidal ecology, we do offshore ecology, we do lab mesocosm work, we do field experiments and measurements, and we also do molecular physiology to try to understand the genetic and the physiological mechanisms that underlie the variations that we see in things like sea urchins, mussels, and algae. Acidic is a smaller consortium of people in Oregon and Northern California, which is focused on coralline algae, which are a major facilitator of other kinds of intertidal organisms. So these are the data, the pH data from the first summer. They range from our sites up in Oregon down through Southern California. At the top is shown the winds. Blue means blowing southward or along the coast. In other words, those are upwelling, driving winds. Red means winds that are blowing to the north, so they're reversals of the usual pattern. And what we show here is for the Oregon coast and for the Southern California coast. So you can see right away that the wind pattern is different for these two regions. And the things to see in these different data traces, first of all, there are intertidal sensors are the thin lines, and the moorings are the thick lines. We didn't have a mooring here, but we did at the other sites. You can see gaps in the data. This is when the instruments stopped working, and we had to get them going again. And these data have been cleaned up. We got rid of nonsensical data. So you can see that there are both large-scale variations, that the pattern we see down here doesn't mirror very well the patterns we see up north, that there are shorter-term variations that are associated with upwelling events, for example. Big declines in pH occur after major upwelling events, and that occurs everywhere. The trends on the shore tend to mirror those in the nearshore water. So the water we're seeing in the intertidal is basically water, the low acid water is actually being carried all the way into the shore. So this is something that was thought to be not to be the case by oceanographers, and what we found is that in fact when low pH water arrives in the upwelling zone, it gets all the way to the shore. Okay, just a little biological work. One of the things we're focused on in the Omegas project is larvae, and we've looked at mussel larvae, and in particular the effects on their growth and shell strength and shell thickness. So we found in these experiments that under elevated CO2, the shells are 15% thinner, and this is a high, like a 2100 level of CO2. This is low. This is supposed to reflect current levels. And this is thickness of mussel shells, larval mussel shells. So we found they're thinner, they're weaker. If you put them in a torturing machine to measure shell thickness, they are weaker. And the biomass is quite a bit less under high levels of CO2. We're also looking at coralline algae. Coralline algae are a major component of intertidal systems, especially in the low intertidal. It's hard to see because of the light here, but 99% of all kelp recruits that occur in the intertidal are found in coralline algae. So they're important sites for kelps to recruit, and presumably their loss would have a very serious impact on the productivity of these locations. We're also doing mesocosm studies, again to vary the levels of CO2, and using various techniques like coloring them with, or dyeing them with calcifluor to look at growth, the effects on growth of differing levels of CO2. A very preliminary result from this investigation is that we found that there's a negative effect. So pH is here, low pH or more acidic water is over here, more normal water over here, and this is productivity on the y-axis. These lines down here are controls where there was no light, and then we had 2 light treatments. And you can see that when they have light, there's a negative effect on growth of the Coralline, that's what the top one is, under high acidic or more acid water. There is no effect on the surf grass, and on the dominant kelp in this system, there actually is a positive effect. They seem to do better with higher levels of CO2, which makes sense since that's what they are growing on. So that's a fairly interesting result, and we are continuing to work on this project. So the biodiversity surveys that I mentioned, these are really important. We actually have two programs that we've done. We've compared two initially, and then we settled on a single one after about the first 5 years of work. And among the things that these surveys have allowed us to do is basically to redefine the biogeographic provinces and breakpoints. For many years, it was thought that Point Conception and Cape Mendocino were the big biogeographic breakpoints along the U.S. West Coast. And our very high density survey shows that the Point Conception one is real, but there also are other biogeographic regions out on the Channel Islands. And then there's also a couple of breaks up here. You don't actually get into the Northern California and Oregon and Washington system until you're north of the Bay Area. And then there's one here from Monterey Bay to the Bay Area, and then one between Monterey Bay and California. So we're learning. I'll just skip over this one, I hope. There. This shows the results of one of my former students' theses, PhD theses. He was interested in looking at how biodiversity was influenced by regional species pools versus local sorts of factors, and how this varied with scale. So these are sampled at across a large regional scale, and these plots are at an intermediate scale, and these plots up here are all quarter square meter plots. And the idea was that if regional, the number of species available in the region is driving diversity, you'll see a straight line relationship. If local processes are having an effect, things like species interactions and environmental stresses, the lines will reach an asymptote and bend over as space runs out and competition starts to be important. And so what he found was that species saturation was the greatest at the quadrat scale, and therefore felt that interactions were likely to be important at this scale, probably not so important at this scale, and that here at the very larger scales, the availability of dispersers is what's driving the diversity patterns that you see. Another use of these data sets was made by my postdoc, Tarek Gouillet. And what he's looking at, he used the mussel abundance data in his study to try to relate the pattern of abundance, which is shown by these red dots here. This is distance from the the site of origin, and then each dot shows sites that are increasing distances apart. And what this is a correlation. So sites that are close together are strongly correlated. As you get farther apart, they actually are asynchronous. And then as you move even further apart, 600 kilometers, they become synchronous again, and so forth. So there's this wave pattern of positive and negative correlation between mussels starting at a particular sample site and comparing subsequent sites. He also wanted to see if this was driven by environment, because a lot of work has suggested that the patterns of dispersal, which are driven by environmental variation, were driving these systems. And what he found basically was if dispersal is limited, in other words, it occurs on that larvae go from one spot and don't go too far, a relatively small number of kilometers, then you get a pattern that shows that through his model that follows this sinuous sort of pattern of first positive, then negative, then positive, then negative correlations. this is how the environment would predict that mussel abundance would change, and there's basically no relationship between the environmental variability and the mussel abundance in any of these cases. So if you use regional dispersal, in other words, they disperse over very large distances, you see no correspondence between the model prediction or the environment and the actual pattern. So this argues for a model of fairly limited dispersal patterns, and it has implications for marine reserves because what it suggests is that you need to scale your marine reserves in relation to these relatively limited dispersal distances to maximize the pattern that you want to achieve in your reserves. Another use of these data sets is that we went to them and sorted all the organisms into what kind of calcifier they were. A lot of them aren't calcifiers, but a lot of them are. And there are different types of groups, different kinds of calcifiers, those that precipitate aragonite, those that precipitate two kinds of calcite, high magnesium and low magnesium. And examples of those are shown here: limpets, whelks, snails, mussels, barnacles, and the coralline algae. And what we've found, if you look at the mobile species like these things, that there tend to be no latitudinal patterns along the west coast. This is the northern coast, okay, and this is the southern coast. There tend to be undulating patterns amongst the sessile species, but also if you do regressions, you find that the high magnesium calcite species tend to be more abundant in the south, and the low magnesium calcite species tend to be more abundant in the north. With respect to marine reserves, PISCO was involved in, again, the design and the theory of marine reserves. Marine reserves were established in Southern California on the Channel Islands in about 2005. They are spreading now up the coast. Here's the set of reserves that move up and down up toward San Francisco Bay. And they include marine conservation areas as well as strict reserves. And that's shown by these different boxes here. And this program has basically worked hand in hand with politicians and managers in California to implement a set of marine reserves that was mandated by laws passed to create the Marine Life Protection Agency, or Act. Data that PISCO has amassed and taken from the literature and also some of its own shows these kinds of effects on biomass, density, size, and diversity of marine reserves. So this just shows the percent change in the various measures in reserves compared to non-reserve areas. And in all these measures, but especially with biomass and density, there were major effects. Species inside reserves are more abundant, they're larger, and they're more diverse. This shows another example looking specifically at lobsters in Southern California. And these are the results from the first few years of the new reserves set up in the Channel Islands, comparing areas outside the marine reserves to ones inside the marine reserves. And you, again, can see that the lobsters tend to get bigger in the marine reserves compared to what they are outside the reserve. The database. I won't say a lot about this. It's a complicated thing to try to store both ecological and oceanographic data. Oceanographic data are generally easy. Ecological are harder because they're more diverse, more different things are measured. And what we focused on initially was to provide detailed metadata. In other words, how did we actually do the different things so that anybody using the data could understand what the data meant. And we spent a lot of time on quality control. Our database manager and another person spend most of their time checking the data streams and making sure that the data are good. We've had quite a lot of hits. This is a publicly available database. A lot of activity on it. People from all different sorts of agencies and private organizations and so forth have downloaded data and used it. We have a user-friendly interface so that you can go and actually it'll generate graphs and other things for you on the different sorts of data sets that you choose, and the data sets are all listed on the data set. With respect to interdisciplinary training, we've actually taught 3 different kinds of courses over the years. The core one was this first one where we trained people in the use of biomechanical, physiological, and genetic approaches to study of their systems. We had a couple times we've run a physical oceanography of physical and ecological oceanography. And we had a policy course which recently has been taken over by the Center for Ocean Solutions in Monterey Bay. We've had quite a large number of students. These numbers are outdated, but they give you an idea of what the numbers are. They move into various kinds of jobs. We still have a large number of students and postdocs. 3 examples are shown here. Laura Patesh was a student in our lab in the early 2000s. She's now working at NOAA and in charge of some climate-related analyses and activities. Joe Tabersi was another one of our students. He became a Knaus Fellow, which Knaus Fellows go and work in the offices of legislators in Washington, D.C., and then we hired him back as our policy coordinator. Anthony Korincic was a graduate student. He's an oceanographer. He was co-advised by me and did his thesis on the mooring data from the PISCO project. And he's now a WHOI assistant scientist at Woods Hole Oceanographic. So what's next for PISCO? We are in our 3rd 5-year plan. We have 2.5 more years left. What are we looking toward the future? Most of our gross domestic product comes from coastal activities, commerce and so forth. That's where most people live around the world. The impacts of humans are greatest on coasts, and that's also where, at least in the ocean, climate change impacts will be concentrated. Large marine ecosystems occur around the world. I mentioned earlier there are 64 of them. This is just one of them, the one that we've been working in. The one here is the Mediterranean large marine ecosystem. One thing I would like to urge you to consider, I know this is being discussed, but you are in a perfect situation with your long, relatively straight coast to establish a PISCO-like entity here, and I know that Gil Reloff is working towards establishing such a program. Beyond 2014, which is when the PISCO grant ends, we have been already working with people in the government and other areas to try to propose the idea of using PISCO as a model for consortia and other LMEs. So we're here, but there are several that are around the U.S. and Canada and Mexico. And it's our vision that each one of these should have its own ecosystem monitoring program, and to do these different things that we do, because we feel like this is a really powerful approach. You can do these things on land too. Here's an example of the nutrient network that was founded by former colleagues who are now at the University of Minnesota, and there the idea is to do experiments in grasslands around the world that involve testing the effects of nutrient additions to these systems. They were particularly interested in productivity and diversity, found some interesting results that actually ran contrary to what much of ecological theory predicted. So I'm almost at the end. Our primary funding has been the Packard Foundation, who supported us right from the start. We've had support from both federal and other private groups, NSF is heavily involved, especially in this omegas project and the acidic project. And I'm done.